Good afternoon, ladies and gentlemen. I'd like to spend some time talking to you about increasing the speed to clinical trials of highly potent molecules. My name is David O'Connell and I work for PCI Pharma Services. PCI Pharma Services was born around about a year ago, and it's an accumulation of 4 companies which you may or may not be aware of. These are Anderson Brecken, Biotech, Penn Pharma and Packaging Coordinates Inc. Now, I'm just going to skip through the corporate slides. If you do wish more information, we are on Hall 3B, Section 16. Okay, so what we specialise in is more molecule to market, so everything from pharmaceutical development to clinical trials manufacturing and commercial manufacturing to clinical trials packaging and commercial packaging, with specialities in high potent molecules as well as some serialization as well. Okay, today I'm going to talk to you, and it's split into 3 parts. The first part is a means of speeding up the clinical trial. So I'm going to present a problem statement and then some resolutions to that. And then we're going on to how we actually onboard high potent molecules. And then thirdly, how to actually perform manufacturing development in a contained manner. So part 1 is the means to speed up the phase 1 pharmaceutical development. So the problem statement we have is pharmaceutical research and development is producing more and more new chemical entities to meet specialized needs such as oncology. The scientists are screening numerous molecules to meet the means of the diseases. And there's a continued demand to actually improve things such as the speed as well as the cost as well. So we want to reduce the drug development cost. We want to reduce the amount of active pharmaceutical ingredients that we actually use in the development and manufacturing, as we know these cost a great deal of money. There's a great deal of competition, so you wish to try and speed up your development to try and get to market as quickly as possible. And the complexity of the clinical trials, so we may have doses from milligrams right up to a gram, so we wish to try and perform the most efficient way of developing and manufacturing these products. So through the clinical phases, as we all know, this can cost billions of dollars or in euros in the case where we are right now. So what the question is there a way to shorten this time frame of drug development and manufacturing? So a traditional way of developing a phase 1 pharmaceutical development product would entail these various aspects. So from excipient compatibility, so where we're actually looking at any gross incompatibilities with the API and the excipients, through to analytical method development, where we're developing a specific method for that API that we can actually detect. Prototype development, so screening numerous different types of formulation and the process involved in that. We have some short-term development stability studies where we're actually looking to see we can narrow down the number of prototypes into one specific prototype. And then we can actually refine that formulation, so we may want to alter the formulation to meet the release profile that we wish to achieve. And then we may want to actually play about with the actual manufacturing process, so we ensure that we have a robust process at the end of the day that can be reproducibly manufactured. Along with this, we have the clinical trials manufacturing, the full ICH clinical stability program, and the QC release and QP release as well. So overall, this can take 9 to 12 months and can cost approximately €500,000, and that is just for the drug development part here. We're not talking about the complexity of the clinical trial or the supplies of that. So a means to actually shorten this would be to use a drug in a capsule formulation, so where we actually dose neat API into capsules. This approach can reduce the overall timeline, It can reduce the financial investment required, minimizes the amount of API required, which then minimizes the amount of API cost, and it reduces the amount of the formulation time and analytical method time. So how does this picture compare to the traditional method? Now, we've still got some aspects that we have to perform, but we're looking at some analytical method development. We're looking at some encapsulation methodology. So this could be either simplistically hand-filling into capsules of neat API, or it could be a semi-automatic process, such as using a profiller or fet-in plate, or it could be actually using a fully automatic encapsulation machine. We still need to perform some development stability, so we still need to ensure before we release the clinical product we have some stability to back up between the API and the capsule shell. And then we have the clinical manufacture and the full ICH stability and QC release. So you can see already that we've reduced the number of tasks involved, which will reduce the overall time plan and also the cost of the development. However, as I was saying, R&D scientists are screening numerous cases. If you look at the example of, say, oncology, the APIs are becoming much more specific and biologically active. This means that we are actually requiring dose strengths much lower and lower. So what we kind of say is, like, for a hand-filling way of manufacturing or an automatic encapsulation, that we can't really go below 50 milligrams. And the main reasons why we can't do that is the length of time if you're an operator to try and fill, say, 10 milligrams into a capsule is very laborious, but also the encapsulation machines, if you put it on there, you get very inconsistent results. So your results would be, all over the place, and that would actually use up more and more API. So it's not a cost-effective solution. So you've still got these very highly active and lower dose strengths of API. So one mechanism to resolve this is to use an Xcelodose. So if people aren't familiar with Xcelodose, it's a specific microdosing technology to fill neat API into capsules. Now, the advantage of this is that you can actually go down to 0.1 of a milligram. So where we've got 50 milligrams is our top on hand fill and on automatic encapsulation machine, we can get down to 0.1 of a milligram. So if you're looking at safety tolerability studies and looking at the dose range that you're requiring for your early phase 1 product, you might have a range from 1 milligram to 75 milligrams as an example. So the Xcelodose provides a means that can fill all the required dosage strengths. The way that it actually works is it actually breaks and builds bridges. So what do we mean there? So the API normally comes in light and fluffy, not very well powder, in terms of the powder flow isn't very well. So we actually use high frequency, specific pore sizes, and the length of time that the high frequency is applied to the dosing head, we can actually specifically or perform a specific method to get down to the very low levels. So at PCI Pharma at Tredegar, South Wales, we actually have two Xcelodose machines. So the first machine that you see here is the Xcelodose 120S. This is a semi-automatic machine, so still quite laborious in terms of the activities that have to occur. We have to manually separate capsules. It goes onto the machine with a specified developed method, fills automatically, and then we have to manually close the capsules before they go into their primary packaging. The machine can run at around about 120 capsules per hour, and it's a full PLC controlled system. So what you can see in picture 2 is actually the presentation that you get. So it's a 10 capsule machine, and you fill it, and as you can see here, it's a traffic light system to indicate if you're within the specification that you've actually applied. The PLC system is also, you can develop the actual method and store the method. You can actually have operators, ones that can actually develop methods, and people that can only run the actual method. So it's a very sophisticated piece of equipment to obtain a very low API fill into a capsule. The bigger brother, which is the Xcelodose 600S, which has been operational at PCI since December of 2015, is the fully automatic machine. So we can actually load capsules. They are automatically separated, automatically filled, and automatically enclosed. So what that means is you can actually get a much higher run rate. So instead of being 120 capsules per hour, we can actually achieve 600 capsules per hour, so a much more effective way. It uses the same PLC system as the 120S, and the methods that are developed in the 120S are actually transferable to the 600S. So on to high potents themselves. Now I've got some statements here. So we have, I have to read this one, a pharmacologically active ingredient or intermediate with a biologically activity at approximately 50 micrograms per kilogram of body weight or below in humans, or a daily therapeutic dose of 1 milligram or below per day. I'll miss out the middle one. The third one is a novel compound of unknown potency and toxicity. So what I'm trying to express here is there is no common definition for a highly potent molecule. There's numerous reasons why it could be highly potent. So a good way of looking at it is all substances are poisons. There are none which is not. So we'll take the classic example of paracetamol. If you engage in 4 grams of paracetamol a day, it's got a therapeutic benefit. If you exceed that, then it can cause some real toxicity problems within the liver. Now, no chemical is completely safe. Now, the analogy that I tend to use here is water. You know, we all need water to actually be a living organism. However, if we excessively consume water, then we have major problems and it becomes toxic to the body. And then one which is more specific to the APIs that we work on, the highly potent ones, is even the most toxic ones can actually be safe if exposures are low enough. Now, why I try to bring that in is in terms of how we actually handle the compounds themselves. So the types of highly potent molecules we work on are APIs with no defined or limited animal-human toxicology data. Now, this is really important for phase 1 molecules because you don't tend to have the full data package at the time that you're actually manufacturing and developing the products. It actually comes along as you go along the clinical phases. APIs with specific modes of action, so it might be something like a kinase inhibitor, which is a very specific mode of action. APIs with no threshold for toxicology concern, or TTC, and this is looking at compounds which are potentially genotoxic. And then there's also molecules which may, generally not be potent, however, they may have a highly sensitizing effect when they're being manufactured and developed. So our answer to this at PCI was to develop a specific facility which took away the need for PPE as the primary source of protection. So you've all seen people working with potent molecules with big air suits on. That doesn't happen in our facility. That's actually our last level of defense that we use for a breach situation. The way that we contain the molecules is to actually use highly contained equipment. So what we mean is equipment which is running under negative pressure, so it almost acts like an isolator in itself. Any movement of powder is either via vacuum transfer or pneumatic transfer or via IBCs with split butterfly valves, so no open handling of powder. The facility itself is run under negative pressure with a single pass HVAC system. And we have the appropriate facility segregation, so we've got specific routes for materials coming in, coming out, personnel in, personnel out, and at the highest risk areas we actually have an additional airlock in place as well. So the onboarding of potent molecules is extremely important. We work in PCI as a CDMO, so we win work from clients. So Our business development team go out and try and conjure up some new business. What we get back on site is either in the form of an RFI, RFQ, RFP, and the kind of information that we're actually looking for from these documents is what is the toxicology that's presented. So this might be preclinical toxicology. What kind of dosage forms are you looking for and strengths? The batch sizes and the method of manufacture, and it's all tailored to actually assess a molecule in terms of its safety, but also can we deliver a service to the client. So we follow a lean philosophy of SQDC, which means safety, quality, delivery, and cost. And these are the actual milestones that we actually go through when we're assessing a new molecule. So in terms of safety, our business development colleagues with the client actually fill out a questionnaire 1. So this is where we're actually looking at any available toxicology data that is there. There's then an internal committee that sits down and we discuss the mode of action or the molecule type, the way that we're actually going to manufacture it, any potential dilution factors of API with excipients in the dosage form, and basically what we can do is calculate an internal occupational exposure limit. And then we actually decide, does this fit with our capabilities? If all these answers are yes and the occupational exposure limit is within our acceptable range, then we can proceed to a proposal. So we're lucky enough, we've won a new project. Now the work has been passed on to operations and the client. And what we now say is, we have a second questionnaire to be filled in, and this is actually between operations and the client, where we go into much more specifics about the molecule, the manufacturing, what type of phase we're going for, the packaging types and such like. And at the end of it, we have a potent passport where we've actually consulted with a third-party occupational hygienist company who have provided a true occupational exposure limit based on the data available. And this potent passport actually follows a molecule wherever it goes. So it doesn't matter if you're working in the warehouse or you're working in manufacturing or in the QC laboratory. This passport walks around with the molecule, and this tells you everything you need to know about it. The occupational exposure limit, the mode of action, molecule type, just so everyone is aware that this is a highly potent molecule. Life cycle management is very important. As I was saying, if we take the example of a phase 1 product, we have to give it a highly potent tag because the data isn't available. However, as you go through the clinical phases, it might not actually be a potent molecule because the data suggests it's not. So the document that we produce has to be peer reviewed, and then it's actually reviewed periodically as new data becomes available. We also have the training and the medical assessment, so very, very important. You know, HSE is our primary drive here, keeping our employees safe at all times. So the employees go through a regular yearly assessment of their health. And also before you can even enter into the high potent manufacturing development area, you actually have to sit through a series of potent molecule training sessions. Which has got an exam at the end, which has got a greater than 90% pass mark. So we do not want any amateurs within our facility. We want highly trained personnel to treat the APIs with respect and also to provide a service that is required. So the third part is to try and accumulate everything we've got here. So we've got an API, highly biologically active. Dose ranges are below 50 milligrams, so we're going to use the Xcelodose. It's a high potent molecule. How do we actually handle it? The Xcelodoses themselves are not contained equipment. So we've actually developed and purchased 2 new pieces of equipment. The first one is called the Xcelohood, where we actually put our Xcelodose 120 within a laminar flow cabinet, which has got a series of H14 HEPA filters, and we can actually perform the relevant task of capping the capsules, filling the capsules, closing the capsules, all within the one unit. And specifically to the Xcelodose, the way it actually measures the amount of powder within the capsules is on a microbalance, a 7-place microbalance, so very, very specific. So we had to ensure that there was no disruption with the airflows of the negative pressure of the cabinet going through and disrupting the balance. The second one, which is the newest machine, is the Xceloprotect, sorry. So this is specifically for the Xcelodose 600S. It is basically it's an isolator, so it's very similar to a dispensing isolator. We've got a complete enclosure. We've got running under negative pressure. We've got HEPA filters in, HEPA filters out. We've got a means to wet down the material when we're actually in a cleaning phase. We've got a very specific materials in and the materials out, and we ensure that the system is actually working properly before we even use it by running a pressure decay test. So where we actually overpressurize the system, watch it decay down to ensure that we're in a safe zone. Again, we had to be very careful in regards to the microbalance on the Xcelodose 600S. So what we have here is the Xcelodose is actually sitting on a floating seal, which then buffers any air movement. So this is a way to, as I say, in conclusion, to try and develop a faster way of a phase 1 solid oral product, which is a highly potent compound. Thank you very much for listening to my talk. Any questions? The maximum fill weight using the Xcelodose is based on the bulk density. So there is no upper limit. So if you've got a size 4 capsule, then you can only fill in a certain amount of material. If you've got a size 0 capsule of the same material, then you'll be able to fill higher amounts. We've filled comfortably into a size 0 capsule 100 milligrams of API. Thank you again for listening to me.